Okay, just let me start our seminar. So, hello everyone. This is Sarin from Kyoto University. Thank you very much for joining our MMP seminar. And it is my really great pleasure to welcome you all and officially start today's session. Okay, Miyoshi-san.
Okay. Hi, good morning, good afternoon, and good evening, everyone. My name is Saroja. I'm co-organizing this seminar with Professor Simon Lee. I'm head of clinical pharmacology and bioanalytics at Pfizer R&D Japan, and I'm also serving International Society of Pharmacometrics as an executive board member. I'm very happy to welcome all of you to this evening seminar. So as you know, today, mathematical modeling is becoming an essential approach in both drug discovery and development. By using models based on biological systems and disease progression, we can better understand human health and help create new treatment more efficiently. So I hope this seminar series will become a great place where people from academia and industry can meet and exchange ideas and work together. Thank you very much.
Okay, thank you. Then we would like to also introduce our nice five advisor. So please, advisor, could you turn on your camera? Okay. Kabashima Sensei, could you turn on your camera? Yeah, thank you. Okay, then let me introduce our advisor. First from the Kyoto University, and the order I just choose the order of the web page. So Professor Kabashima, could you start?
So my name is Kenji Kabashima from Kyoto University. I'm a professor of Department of Dermatology, and I've been interested in the pathogenesis of inflammatory skin diseases, and mathematics is not my specialist, but I'm interested in this session. Thank you very much.
Thank you. Then next is Professor Yanagita.
Okay, so hello everyone, and thank you, Seiri-san. My name is Motoko Yanagita from Kyoto University. I'm the professor of the Department of Nephrology and interested in the kidney disease and the repair mechanism of the injured kidney. Recently, I started the collaboration with Seiri-san and realized the power of mathematics, but I'm quite outsider, but still I'm interested in today's lecture. Thank you very much.
Thank you. And then next is Professor Kawamoto.
Hi, this is Hiroshi Kawamoto. I'm a director of Institute for Life and Medical Sciences. And now, so our group is developing a new type of cell therapy in which T cells are generated from pluripotent stem cells like ES or iPS cells are used to treat leukemia or viral infections. I myself is not good at science, mathematics, but I'm feeling so like some to make a model of in cell therapy that is like a pharmacokinetics. So still how cells behave or the dynamics of cells are not so much established in terms of mathematical models. In that sense, I'm very much interested in this seminar. So thank you very much.
Thank you. And then we are going to the advisor from Pfizer. So Dr. C.J. Musante, please.
Thank you very much, Sarah, and thank you for organizing this session today. I'm CJ Musante. I'm Vice President of Scientific Research at Pfizer and most recently Global Head of Pharmacometrics and Systems Pharmacology. I am a mathematician by training, and it's so fabulous to be here with this panel who are interested in applying our techniques to really important questions in drug discovery and development. So it's my pleasure to be here. Thank you.
Thank you. And then Dr. Kawai?
Hello, everyone. I'm Nori Kawai, Clinical Research Advisor at Japan. So my group is responsible for developing the clinical development strategy in Japan. I believe that the QSP is an innovative approach for drug development. So I'm looking forward to today's lecture. Thank you so much.
Thank you. So then we will start the main session. So please, Miyoshi-san.
Okay, so I'll chair a session of a presentation by Dr. Saroja Ramanujan. So I'd like to ask Dr. Shigemu-san to introduce Dr. Saroja. Dr. Shige, please.
Thank you. So it's my great pleasure to introduce my collaborator, a former colleague and good friend, Dr. Saroja Ramanujan, to this ETH seminar. Dr. Saroja is an expert in quantitative and translational and systems modeling in biomedicine. After completing her Bachelor of Science degree in engineering and PhD in chemical engineering, she studied tumor interstitial and microvascular transport as a postdoctoral fellow in my backyard at the Massachusetts General Hospital and Harvard Medical School. She has since spent 25 years in biopharma companies supporting drug and diagnostic development, including 11 years initiating and leading the translational and systems modeling group at Genentech, which in my opinion is one of the best in the industry. She currently serves as an independent consultant on biopharmaceutical drug development across therapeutic areas and modalities. She has published extensively in the field, including a book chapter that we both co-authored very recently on this topic. She's also served on the editorial board for the Journal of Pharmacometrics and Systems Pharmacology. And if you're new to the field, I suggest that you check that out. It's one of our leading journals. And she's also a guest lecturer for multiple courses for the University of California at San Francisco in artificial intelligence and computational drug design and development in the master's programs there. Last but not least, Saroja and I, 10 years ago, started the QSP Special Interest Group as part of the International Society of Pharmacometrics, along with our colleague, Michael Zager. She's also a fellow of the International Society of Pharmacometrics and a recipient of one of our most important awards, the Innovation Award for contributions to quantitative systems pharmacology. So thank you, Saroja, for joining us tonight. We're really looking forward to your seminar.
Thank you, CJ, for that kind introduction. Thank you, CJ, Drs. Miyoshi, Dr. Lee, everyone for the invitation and the opportunity to present to this exciting international collaboration organization. It's a topic close to my heart. The idea of not just a specific area of pharmacology, but the idea of marrying modeling, mathematical modeling, and biology more broadly. I will share my slides. Is that okay? Is this a good time to do that?
Yeah, of course.
Okay, I will go ahead and do that, and then we can get started. And let me put it in presentation mode. Stop, continue to share. Can you guys see this? Can everybody see? Okay. Well, I guess it's August 18 for you. It's still August 17 for me. So good morning to you, or whatever time of the day. I know we're spanning multiple time zones. But my talk today will be on more focused in the area of quantitative systems pharmacology for drug development. Of course, mathematics in biology and medicine spans much more than this, and I'll touch on a few other areas very briefly, but this is my area of expertise, and I thought it would be fun to discuss that with you. Let's see. Okay, so we have this term called model-informed drug development, which we now use this approach of model-informed drug development throughout the drug discovery, development, and approval space, and even post-approval. This can be in research and discovery for looking at new biology, for trying to identify targets, designing drugs. This can be preclinically and translationally as we do in vitro or in vivo studies on specific drugs and look to translate that behavior into the human for safety, efficacy, and trial design. In the clinical space, there is certainly trial design for dose, for regimen, for biomarkers, for patient stratification, to look at combination therapies, to compare efficacy, to make predictions in all these areas. Models are used. And finally, for approval and post-approval, modeling and modeling results are often used for dose and regimen justification, to address behavior in special populations, maybe organ impaired or pediatric or other special or prenatal populations or pregnant populations. And then post-approval, we can look at things like population and indication expansion. We can look at things like modifications to approved doses and regimen. And in all of these areas, modeling plays a role. And even beyond this, I should say that even in we have health economics modeling that can inform insurance and payer decisions and various applications in biology and medicine. Within drug development, some of the common model-informed drug development approaches are listed below, and I'll go into those in a second. But one of the things that tends to be the case is earlier on, when you have less data, you are more reliant on understanding and knowledge about the biology and the mechanism, and possibly hints of data from this study or from that animal model or from this in vitro study, and you're trying to piece things together biologically or based on mechanism. As we go further and further and generate more data on a drug of interest or pathway of interest or a population of interest, we begin to enable the use of empirical or statistical approaches driven by that accumulating data. That does not mean we stop using mechanistic approaches. And that doesn't mean you can't use some of the empirical approaches earlier, but this is a general trend. That said, I will be talking today about examples in the systems pharmacology space, all the way from biology through late stage. So you will see that mechanistic models can be used right through. So I thought I would highlight some of the example methodologies that are well known in drug development. I'll start with quantitative systems pharmacology or toxicology as one of my specialties, which is really looking at biological mechanism-related models that look at a biological system, maybe a disease, maybe a pathway, maybe a set of drugs, all with the biology around them. And those tend to be very mechanistic. There's another form of mechanistic models which are really more focused on the physiology and how that influences the pharmacokinetics and the downstream pharmacodynamics of drugs. And that is called physiologically based PK or PBPK, and it can extend into pharmacodynamics, so PBPK, PD. And so we're looking here at physiological compartments, different tissues, flow of blood, of lymph, of drug, how it moves, and then you get to really look at target tissues. You might say, what's going on in the lung? What's going on in the liver? As we talk about preclinical and translational PK/PD, this can be either mechanistic or empirical models, which are focused on a drug and how it influences, how its dynamics are characterized and how it influences the dynamics of a biomarker. But it's usually very focused on a specific drug and maybe one or two biomarkers. When we go to population PK and population PK/PD, these approaches are used clinically, typically, when you're looking at the behavior of the pharmacokinetics and possibly the downstream biomarkers in a population. And you want to characterize the mean and the variability around that to make decisions on dosing and understand the exposure patients may get and the implications of that drug exposure. Of course, we then go on to exposure response, where we connect that exposure statistically to some indicator of response. It can be a clinical efficacy marker, it can be a biomarker such as tumor regression, but it is a statistical relationship where you look at an exposure metric in a population and connect that to the response. There are a few other broader methodologies that are used, and one of them is model-based meta-analysis. This is when we use a model, typically a statistical model, to integrate and marry data and learn from data from different drugs, for example, or from related diseases in a common drug family. It's however you define your area of interest, you're doing kind of a meta-analysis, but you're doing it in the context of a statistical model. I should note that in a sense, this can be done in mechanistic disease models also. You can do a model-based unification of different drug effects and compare those in a mechanistic model. It's not the most common way, but in systems pharmacology, we are often doing this. We don't call it model-based meta-analysis, however. And finally, disease progression models. These really describe the natural progression of a disease, as well as the effects of treatment. And so disease could be biomarkers, it could be clinical endpoints, etc. And these models are often statistical functions of, baseline biomarkers or baseline patient demographics. And they could also be mechanistic disease models that follow the trajectory of disease progression and biomarkers over time. And these are one of the places these can often be used is in looking at control arms of studies where you want to say, well, I expect my drug to do this, but it's superimposed on a natural progression of disease. How would an untreated or a control-treated population look on the progression of that disease? How would a treated population look with progression of disease and comparing those? You can think of Alzheimer's, for example, as a case where progression is really one of the metrics. Of course, as I said, these areas are being increasingly adopted. And as an example, I thought I'd show a slide from an FDA review on the evolution of model-informed drug discovery and development at the FDA. And without going into all of the detailed bullet points and everything on this slide, I think we can just look at this main arrow and understand the general trajectory of MIDD, as I'll call it from now on. in drug development. So early on in the late 90s, it was considered the early days of MIDD, and the FDA started looking at things like POP-PK and PK/PD modeling. In the beginning of the 2000s, there was a rapid growth in approaches, and you started to see more of this being incorporated into the FDA and its discussions. In 2010 to 2020, their terminology is it's approaching mainstream. Many of the approaches were POP-PK/PD, for example, population PK/PD, for example, or PBPK for certain areas was becoming very common. And other areas were just emerging in terms of regulatory interactions, things like quantitative systems pharmacology. And then finally, where we are right now is these are really integrated throughout drug development. Regulatory bodies are definitely looking at these, and we're now starting to see more coming up in the artificial intelligence, machine learning, real-world data, real-world evidence area. So there has been this trajectory. And of course, what we see in the regulatory space will always come, is a reaction to what is being developed in research organizations such as Kyoto University or wherever. And as they see these approaches, they are looking at how they can adopt them, and they will also start doing their own research in them. But as I said, my expertise is in an area called quantitative and systems pharmacology. So this talk will focus on background, examples, and future directions in this area. So before I go into this, I want to talk about some of the pioneering efforts we saw that led to the field of quantitative systems pharmacology. And I would just call this biological systems modeling. We used to just call it mathematical modeling of biological systems. And some of the very well-known models is a researcher named Guyton had a very large-scale model of blood circulation. Dennis Noble had developed a model of the cardiac cell and the ion channels, and that has now moved on and become the very core of a lot of work that's done in cardiac modeling. A engineering leader named Doug Loffenberger did a lot of work in receptor and ligand dynamics. How do receptors and ligands bind? How are they sorted? What is the implication of this basic interaction that we know is so important in medicine? And then another lab, for example, is the Rakesh Jain's lab, which is where I did my postdoc, and he did a lot of early mathematical modeling in tumor microphysiology, microvasculature, and tumor transport. So these are some, by no means all, of the areas that really led to this field of quantitative systems pharmacology. In 2011, a group of scholars and very well-known leaders in this space came together at an NIH working group and drafted the NIH white paper on quantitative and systems pharmacology. And this really kicked off the idea of quantitative and systems pharmacology as a field. If I say, what is this space? It really integrates some different background areas, looking at biological systems, using mathematical formulations, engineering, computational approaches, and also thinking about PK/PD and pharmaceutical sciences, and marrying these three areas under this umbrella of QSP. So there's no real clear, crisp definition of QSP, but more descriptions. This is one I like: quantitative analysis of the dynamic interactions between drugs and a biological system that aims to understand the behavior of the system as a whole. Most important to me here is behavior as the system as a whole and the drug and the biology. The dynamics is often there. I would point out it doesn't have to be fully dynamical. It doesn't have to be fully mechanistic. It just has to have these features, really. Some of the common features you'll see in these approaches are a mathematical representation of key biological relationships. The form of this math can be anything. It could be differential equations. It could be statistical equations. It could be whatever makes sense given the data and the model you're working with. One very critical point is these models prioritize capturing the biology needed to answer the questions at hand over being as parsimonious or as small as possible. Here, if you need to be bigger, even if you're sacrificing the ability to know exactly the parameter values, etc., it is okay. It is more important to capture the relevant biology. And some of that relevant biology are feedbacks, crosstalks, and redundancies that we know biological systems have that can significantly impact the downstream effects of a drug. Often these models integrate diverse data. They are not necessarily based on one clinical trial, because that knowledge of these mechanisms and these different pieces of the system come from different experiments and different studies. There is a representation of the pharmacology of relevant therapeutics, and critically, the ability to use such models to make quantitative predictions under simulated or hypothetical conditions. So I like to draw this in the following way. We have all of these different pieces of information. We integrate this in a kind of a map of disease, which is, or a pathway or whatever you're studying, which is characterized by some form of mathematics, often differential equations, but not always. And then we can simulate that model to make predictions. And when I say it doesn't matter what kind of math, it could be mechanism driven, where we really build it from the mechanism up. It can be data driven, where we have a large set of data and we deduce or we infer mechanisms from it and build that kind of a model, or it could be a mix of the two. And there's a diversity of mathematical approaches, different kinds of differential equations, agent-based models, logic-based models and networks thereof, statistical systems network models. These models can be disease platforms, pathway models, toxicity models, and the size is really highly variable. You can go from a handful of states to thousands of them, thousands of variables, so a large range of size. And as I said, the biological detail depends on what you're modeling. It could be that you're really focused on an intracellular pathway, or it could be that you're looking at a tissue and the underlying cells and intracellular biology. You may be looking at different species and different subpopulations, or you may be looking at a broad population overview. So really, it depends on the question. And as an example, I'm showing you 4 different, very different QSP models. It could be a pathway model of, here it's the PI3K pathway and receptor ligand binding that drives it. Here in panel B, this is a large-scale disease model of rheumatoid arthritis as a disease. On the bottom left is a mechanistic model that marries PBPK as well as representation of biology in the liver to look at drug-induced liver injury. And in the bottom right, this is actually a statistical model where it making statistical but inferred mechanistic relationships between various biomarkers in diseases such as cardiovascular disease or diabetes, etc. That was from a company that used to exist called Archimedes. And as I mentioned, QSP is one of the areas that we are seeing more of in the regulatory space. Without going into a whole lot of detail, I'll just point out that submissions to the FDA with QSP have gone up exponentially since they started to show up in the early 2010s, and this was as of 2020. So I thought now that you have this kind of bigger picture overview of QSP or what it is, I could walk you through some examples of how it's been used throughout the research and development pipeline. So I'm going to walk you through from this biology space through the late stage, giving you examples from the literature that you would be able to find out there if you wanted to look into them in greater detail. But this is more of a snapshot of these different examples. And as I give you these, I think what you need to keep in mind is, when do you need QSP? There were all those other approaches, but what you'll see with these examples is often they're considering or extrapolating to a new or different scenario, maybe a new target, a new compound, a new species, an untested dose or regimen, an untested population or combination, maybe a new indication, places where the data isn't so rich that you can just build a statistical model and have confidence in it. It's often used when you need to decode the biology. Why do we observe this behavior, and what are the implications of this biology? And to test what would happen in a very specific biological scenario, and how, oh, well, if cells were doing this, or if a patient's liver was functioning like this, what would be the prediction that way versus if it was this kind of biology underlying the observation. And finally, for novel biomarkers and diagnostic. So can we propose a priori or very early on some potential new biomarkers or diagnostics that can help stratify our population or predict response? So before I go into all these examples, I thought I would walk you through one. It's not a brand new one. It's from my group from, I don't know, probably close to 10 years ago now. But I think it walks through many of the pieces of QSP so that you understand a deeper application of QSP, and then we can talk at a higher level about different examples. And so this was a QSP model of what is called a T-cell engager. It's an anti-CD3, anti-CD20, T-cell engaging bispecific antibody. One arm is anti-CD3, one arm of the antibody is anti-CD20. I'll come to that in a second. It's used for B-cell malignancies. And this particular effort was started in the preclinical phase. We went on to use it to look at the phase 1 dose regimen, to support the recommended phase 2 dose regimen, and to propose predictive biomarkers. And it has gone on to be adopted to many other applications, but I'll walk you through these. So this molecule that I'm going to talk about is called mosunetuzumab. It is a full-length IgG antibody. One arm, the anti-CD3 arm, binds CD3 on T cells. The other arm binds CD20 on B cells. When both arms are bound, it redirects that T cell to be activated and kill B cells, malignant or otherwise, anything that expresses CD20. It has with it a safety concern, which is when you activate T cells, they can release cytokine that can start a cytokine cascade, and it can lead to a very severe safety event called cytokine release syndrome. Grade 1, acceptable, maybe it's a fever and some chills, etc., can be handled. Can go to grade 2, grade 3, grade 4, and even death. So this can be a very significant safety concern. Now, when we were developing this molecule, we had a little bit of precedent knowledge from a molecule from Amgen called blinatumomab, also known as Blincyto. This molecule is similar that it binds CD3 on T cells, but CD19 on B cells. And its format is very different. It's this little BiTE molecule here as opposed to a full-length antibody. It was already approved for an indication called ALL, but they had started to study it in the same indication we were interested in, which was non-Hodgkin's lymphoma and a specific kind called DLBCL, diffuse large B-cell lymphoma. But we could not just use what they have done for this molecule directly for our molecule. It was a different target, CD20. It was a different format. It was a full-length antibody with very different PK, and it hit different subsets of B cells. So we said, can we use the limited public data from blinatumomab to somehow inform what will happen with our molecule? Well, first we created a model of what our molecule does. We used basic knowledge of immunology, in vitro data on T cell activation and B cell killing by the drug, and primate PK/PD, so PK with multiple cycles and how PD or cell counts may change in the blood with different cycles of treatment. And we built this mechanistic model, which I'll walk through in a little detail. Here you have T cells. They get exposed to drug, they become activated, they proliferate, they can release cytokine, they may die, they may travel to the bone marrow, lymph node, spleen, or to a tumor. Over time, they become deactivated, and they can enter a resting state. Meanwhile, B cells can come out of the bone marrow and traffic, and they, when engaged with these T cells, can be killed. That's pretty much what we were modeling. And we used basic differential equations to do this. And this was based on first principles, measures from animal, measures from that were reported in literature and in vitro and in vivo data. And we did that not only in the blood, but also in a lymph node tumor, a lymph node and the spleen and the bone marrow. The tumor is only in the clinical context. We were looking at a lot of different things with this, including IL-6 production, tumor regression, B cell depletion in all these tissues, and systemic cell numbers. I'm not going to go through what all of this is, just that we looked at different biomarkers in the blood and different measurements in the spleen and the lymph node. We had, did I miss a slide? No, here. We had data from cynomolgus monkey. We made sure our model could capture that data, and we validated it by predicting other data in the cynomolgus monkey, and we felt we had a good model of what this drug was doing in the monkey. But how do we make it a human disease model? Well, we had that data I mentioned from blinatumomab. So we first took our model, we made it human by changing its physiology. We added a DLBCL tumor mathematically, a compartment that was proliferating quickly like a tumor. And we created a bunch of virtual patients based on those original virtual monkeys translated. We then selected out virtual patients that gave us a waterfall plot of tumor response that looks like the clinical data for blinatumomab. We also made sure that that virtual population of patients that gave us that waterfall plot also reproduced the cytokine data they reported, whether they were using a flat dosing regimen of their drug, which means the same dose repeatedly, or a step-up dosing where they start low and then go higher and then go higher. Why are we looking at these dose regimens and why are we looking at this at all? Is because if you look, this is IL-6 I'm showing you. This is a cytokine that is closely related to this toxicity of cytokine release syndrome. While you want this tumor response, that's something you want, IL-6 release is something you don't want. And what you can see is when they went with the flat dosing, they got repeated bumps in the cytokine. But when they went with step dosing, it went right down. And we wanted to know if we could take a similar approach and what doses we would need to look at in our drug to achieve good responses, but mitigate the cytokine levels. And so we looked at a few different approaches. What if cycle 1 and cycle 2, we just dosed on day 1 and went for 3 weeks between? That would be flat dosing. What if we fractionated the dose in the cycle 1 into 3 even fractions before going to Q3W dosing? What if we did a step-up fractionation during that first cycle? And do we need 3 doses or 2 doses during that step-up? So we simulated all those in our virtual population. And what we found was that as you go through these different approaches, we would reduce the cytokine peaks. So the first cytokine peak would go down from, say, 670 or 570 there all the way to 304 if you could, and even lower. And then the later cytokine peaks would stay low, even while the tumor growth was not being significantly affected. So you could step up in that first cycle. As long as you get to that dose by the end of the first cycle, you can still have response. So based on this, we changed our dosing regimen for phase 1B. Phase 1A, we had just gone with this non-fractionated approach, but we were seeing a lot of CRS, a lot of cytokine release, very problematic. And when we switched to this, we really stopped seeing bad CRS, just grade 1, some grade 2. We never reached a dose-limiting toxicity. This drug went on to be quite efficacious and safe and is now approved for this indication and is being looked at for other indications. In fact, what our predictions were quite accurate for the range of IL-6 versus that first dose. And we also, our prediction that this step fractionation would reduce later cytokine peaks was correct. I would now like to go into some of these other examples rather than just boring you with that one case study. So I'll go a little faster now, but I'll try and be clear. If you look at the biology and target, one of my favorite applications where systems modeling can be used is to help identify and evaluate new targets. So whether it's that rheumatoid arthritis platform, which was used to do a sensitivity analysis of what different parameters and nodes in the model, how important they were to clinical outcome, we could look at 15 targets and rank them by their ability to change the clinical biomarker or the clinical endpoint in silico and say these are good targets to look at versus not as good targets to look at. We did this for a company called Organon, which was subsequently acquired by Merck. Another company called Merrimack, for example, I published a paper on how they identified a very specific way of targeting the EGFR family of receptors in order to suppress tumor growth. If we go on to one of the areas I worked heavily in, which is optimizing a drug and looking at preclinical translational work, here I'm giving you an example out of our group where we were working with a biotech on a molecule which was meant to bind the IL-15 receptor and stimulate in an agonistic way and stimulate cell proliferation. Very complicated because it binds to that molecule, stimulates the cell to proliferate, which only increases the amount of cells around to bind that molecule and sink it out. So it had a very complicated PK/PD relationship. And first, there was a model that was developed by a CRO working with the biotech to really optimize the affinity such that they would get expansion, but not so much so that it would suck out all their drug. And then we went on to say, what if we made a more complicated model to really look at the dynamics in the cyno of multiple cell types? And we could then look at the impact of different dosing regimens on influencing the pharmacodynamics in the different target tissues, both for safety and for efficacy, depending on the tissue you're looking at. So this helped inform both the drug design, the CRO work, as well as the preclinical translational work we did using the cyno model and trying to predict how dose regimens would be, what effect they would have in the human. Here, again, from that Mayo group, there's some work in the translational and phase 1 first in human dose that is interesting. One of my colleagues named Iraj Hosseini looked at, again, one of these detailed mechanistic PK/PD models that looks very carefully at cell expansion and binding, etc., to look at how a FLT3 ligand molecule would lead to dendritic cell expansion in a similar way to what I showed you before. And they really used that in this case to justify their first in human dose, benchmarking off of other molecules with similar mechanism. A very different kind of example is in a publication we have on asthma and the full asthma disease model, looking at various cells. So this is very focused on a few cell types and the receptor ligand interaction. This asthma disease model is really looking at various cell types, various cytokines, various interventions it's been built for. But then we could say, well, we want to look at if we had a JAK inhibitor, or an inhaled JAK inhibitor, which was inhibiting certain cytokines downstream of JAK, how would we predict the dose would influence a specific biomarker called the exhaled nitric oxide level? And so we modeled that and were able to help inform what would be a feasible or a required dose to get suppression of that biomarker and that pathway in human asthmatic patients for an inhaled therapeutic. So this is very much in that space of translation and first in human dose, a place where systems pharmacology is really quite valuable. This is an interesting example in the system toxicology standpoint, where we looked at a detailed model of how PI3K inhibitors act on many functions in the gut to possibly, and different PI3K isoforms, to contribute to intestinal inflammation and drug-induced colitis by PI3K inhibitors. And this was very interesting because we modeled the many different drugs with many different profiles of inhibiting these. and showed how those profiles of inhibiting these different isoforms, as well as the differential dosing regimens, which allow the epithelium either to continuously be insulted and damaged or to recover after an initial damage before the next dose, how those different drugs and dosing regimens would lead to colitis. And that helped us design our next generation molecule, which actually was recently approved, called inavolisib. This is an example in the clinical space, a very nice example from Pfizer, from some of my colleagues here, where they developed a model of COVID infection, and they subsequently used that model to explore the dose regimen for different patient populations, where internally and externally, there was a question of whether they would need to dose Paxlovid for 10 days or whether they could get away with 5 days. The QSP model that you see here showed that a 5-day duration would be sufficient for the patient population of interest for their pivotal study. And if they had to include a 10-day arm, that would have added at least 6 months to their development. That said, they went with the 5-day, and then they also said maybe that would be different, for example, maybe for the immunocompromised population, if you depending on whether you're looking at the normal population, the immunocompromised population, and whether you're looking at viral load or the viral reduction, you could consider a 10-day, but the key piece was that a 5-day was sufficient for the population of interest, and they went forward with this, and it was successful. And as you know, Paxlovid is a 5-day treatment course now. So with that, I think I've walked you through examples from early to late development, but there are growing and emerging directions for QSP. And some of these are in that book chapter with Dr. Musante that she just referred to. And some of them are looking at novel modalities of drugs. I've touched on some of those bispecifics or cell agonists that lead to proliferation. I'll talk a little more about this now. And also looking at more in silico trials for regulatory purposes. Moving forward, we can start thinking about what are called novel approach methodologies or novel assessment methodologies. These can be in silico, and they can also be these complex in vitro systems, which reduce the need for animals and maybe have better human type tissue or human type behaviors. And you combine them with the QSP model, it's a very potent approach. Also, we believe there's more and more to be done in biomarkers and precision medicine. And I think you have a speaker coming today for you, speaking about precision medicine. And some common threads are hybrid and integrated approaches for these applications and increasing efficiency and adoption. So a few of these examples, here is one with that novel approach methodology with everything highly integrated here. I thought this was a lovely paper by Adwati et al. In Molecular Systems Biology. They used they took patient-derived biopsy. They used that and tried those, treated those cells with different drugs in vitro. Based on the responses in vitro, they could recalibrate a systems model of signaling pathways to identify which nodes in how that person's tumor was wired, what are the strengths of these different pathways, and predict what would be the optimal treatment combination for that patient. So this is a nice combination of this complex in vitro system, or quite an advanced in vitro system, with or I should say ex vivo system if you want, with a systems model. We heard one of our advisory board here speaking about cell therapy and characterizing cell dynamics with mathematics potentially. There have been two very nice papers. One was looking at how it also looked at cell dynamics, but it focused a lot on CAR-Ts, on how some of the RNA expression pathways were better indicators of response than just cell dynamics. Another paper, this one from my group, looked at modeling the cell dynamics in detail from the different subsets. This was in a TCR transgenic T cell therapy. And here we created individualized virtual digital twins to each clinical patient, because you know that this cell therapy is so individualized. The cells are different, they come from each patient. What they get is different in terms of the mix of cells, how many are highly differentiated, how many of them are more stem cell, and that highly influences the patient response. So we created digital twins and showed that, hey, for each of these individual twins, we can start to explore for that patient what would be the ideal composition of cells to give them. What I would love to see is marrying these approaches, taking this kind of individualized patient approach, but marrying in that individualized patient's omics signatures into the model. And then you could really start to think about how should we treat these patients based on their individual biomarker profiles. And also, you'll start to see more integration of quantitative approaches. It's already started with the inhaled drug, for example. We look at inhalation and make a PBPK model, feed that into a systems model of disease, feed that into an airway fluidics model, feed those together into a statistical model of exacerbation for asthma. And there you have many things together. Similarly, you can think of marrying data-driven and omics data to create causal systems models and really combining different methodologies and data sets. Of course, one of the areas we're all very interested in is how to integrate with AI. And I see a lot of opportunities. This is just one diagram, shows how AI and ML can really interrogate the data to give us ideas of what's important to look at in a systems model, and maybe to infer networks and look at important targets. The other way around, that mechanistic and physiological information out of a QSP model, that we can analyze that with machine learning approaches to look for biomarkers predicted by models. And of course, all of this together can be fed into the drug discovery, development, and the overall R&D process. So just to end, my vision and that of many of my colleagues in the field broadly is that we have an integrated quantitative medicine approach for pharmaceutical R&D. Here we have various non-clinical data types, and we also have patient data. All of this can be fed into different quantitative approaches, a marriage of different quantitative approaches, enabled by a data and model infrastructure, automation, interoperability, etc. And based on this quantitative analysis of the data we're generating and accruing, we can improve the non-clinical systems and experiments, and we can also help design the clinical trials and inform regulatory interactions and personalized medicine by way of these precision medicine and digital twin approaches. And with that, I'd really like to thank you all. I'm missing my acknowledgement slide. I apologize, but I think there's too many people to possibly acknowledge. Certainly my old colleagues from Genentech who did much of the work. There's much of this work was also from other groups, including Pfizer's, including Applied BioMath and various others. And I hope this has been an informative talk for you, and I'm happy to answer questions.
Thank you very much, Dr. Saroja Ramanujan. Your great talk. So now I'd like to move to the Q&A session. So during the Q&A session, advisors and general participants will take turns asking questions to Dr. Saroja Ramanujan. And first, so I'd like to invite CJ to ask the initial questions. Afterwards, general participants will have an opportunity to ask questions. And then, Dr. CJ, please.
Thank you very much. So, Saroja, thank you so much for a wonderful presentation. The last slide that you hit on was really where I think the future is also going in quantitative medicine, and was wondering in terms of the FDA's recent formation of the Center of Excellence in Quantitative Medicine and the interest in this group is many academics as well as industry people on this call, and many in a hospital setting. How do you see, or where do you see the best opportunity for us to collaborate with industry and academics and hospitals to really realize the full value of quantitative medicine, MIDD in quantitative medicine?
Yeah, certainly I've talked a lot about drug discovery and development, and certainly the clinicians, the MDs, et cetera, are centrally involved in that. However, I think there's a lot more that can be done. We can, and there are folks working in medical centers, in the Memorial Sloan Ketterings or the MD Andersons, doing this kind of modeling hand in hand with their clinical colleagues in those settings and influencing maybe not drug discovery and development in that context, because you still need to go through regulators to get, but maybe just how they think about disease and how they treat and how they interpret that and exploring hypotheses. And one of the things that I know the FDA was trying to do with their Center of Excellence around quantitative medicine, and that I think is very important, and I see in this audience here, is this cannot be a conversation of modelers and drug development teams only. It really has to be a place where the quantitative and the medical come together and communicate clearly with each other and understand each other. So I think the big opportunity is there. It could be for personalized medicine, exploring that area. I think that's a big one. It can be in trying to understand the clinical data they are generating and putting forth biological hypotheses together, the MDs and the quantitative people together. That's another great area in quantitative medicine that the clinicians and certainly in the drug development, but I feel like that's happening, but I think we really need to get the MDs and the modelers together even more, and the researchers, the biological researchers, certainly, because those ends are the ends that are hardest. In that core of drug development, we seem to have the MIDD, where the FDA is involved, where the EMA is involved. It's happening. Those two ends, I think, are really a ripe opportunity.
Thank you.
Okay, now we are going to some question from general audience. So general audience, if you have a question, just use the Q&A button in your bottom of your slide. You can find it easily. So we are getting the questions again. Then okay, I will just pick up one. Is the Okay, so patients Abasaki. If you want to, you can just turn off your camera and turn on your audio, and you can directly ask to your questions to the speaker.
Okay, thank you so much for the very informative presentation. And so my question I put in the Q&A was, the whole time you've been talking, I've been wondering about reproducibility of models. And I mean, you've been talking about, I got this contract, this request from this company and that company and blah, blah, blah. And I'm wondering, do you have to go through the modeling process from scratch each time you have such a request? Or do you get to use old models, or at least parts of old models? Because I'm thinking if you have to do this from scratch all the time, then it's going to be challenging, and it's going to be hard for novels like us to really do this on our own if we can't use anything that you have done, with all of your experience. So how does it really work with reproducibility? Thank you.
That's an excellent question. And in fact, there have been papers that have looked back at QSP models or other models to look for reproducibility. Can they implement the model? Can they reproduce the results in the model? And the findings are really disappointing. Many of those papers are not reproducible. I think any of us who has sat with a paper and tried to do it, we know we end up having to email the author and go, wait a second, and then we get the help and maybe we get it worked out. So what we have really tried to do, and we try and publish as much as we could at Genentech. I know that CJ has published. The examples I showed you, they were all published examples. In most of those, except for one that I know of from us, and I don't know about some of the others that were from, we have included the code. And what I mean by including the code I don't mean including printouts of the code. I mean including the actual executable files such that you run them, they reproduce the figures. That example, the T cell engager example, and I've left Genentech. I don't work there anymore. But I took those files, I've re-implemented that model and modified it for other clients' needs moving forward. So I think that is absolutely critical, is sharing as much as possible. And the regulatory, that is in our interest to each of our individual interests, not only because we make it available to each other, but when we publish and it's gone through peer review, the regulators can look at it. They look at it with that same scrutiny. They know it's gone through peer review. They feel more comfortable about looking at it in submissions, etc. This is one of the stories where there was a bone model that was a model of the bone that was published many years ago. When a sponsor came to the FDA with a new molecule, the FDA took that model from the literature and looked at how would that molecule, what would the model say about it? The very fact that it was out there enabled the regulators to look at it. It also enables you. So I think not only publishing it, but publishing it in a way that it can be fully reproduced. Another piece that we worked on a lot at with my old group was either software or methodologies that are robust and that allow dissemination of the model in common language and common approaches so that they can be shared. So I think all of those areas are really ripe for improvement, and I think we need to see more of it.
Okay, thank you very much. The next turn is from the author advisor. So I would like to invite Professor Yanagita to ask a question.
Okay. Yeah, thank you very much for your great talk, Professor Ramanujan. And I learned a lot from your talk. And I have a very naive questions. As a nephrologist, I know that the kidney contains a large number of cell population. However, the crosstalk between these cells is not yet fully understood. So when creating mathematical models, how should we determine the appropriate number of cell types to include, especially when the experimental data supporting this decision is not perfect?
It's a very good question, and it is one of the I would say, is the hardest part of building a mechanistic or a systems model, is what is the scope? What do you include and what do you exclude?
Mm-hmm.
And that is not a prescribed approach right now. You are looking at when you think about a specific application, you have to say, what does the literature tell me are important pathways? What cells might be like, is there a cytokine? Is there something that is heavily involved, and what cells might be producing it or might have receptors for it? So those kinds of questions. What if I am working in a context of with the MD or a researcher? I will ask them, in addition to this literature, do you have hypotheses? Do you think so? And then you know what? You try. And you look at the prior literature. There is, there's CJ's group. There's groups out of Georgia, University of Georgia, etc., that have done a lot more kidney modeling. I have not done kidney modeling myself, but I would look at that literature and see what they have modeled. What do kidney experts or kidney on a chip type things look at? What data is there? And then you make a first effort. You say, this is the scope I'm proposing based on what I know and what we know and what we hypothesize. You might find there are pieces in there you didn't need, that they do nothing. Okay, fine. Later on, those are pieces you can either eliminate or just not spend as much time looking at later on. You might find that the pieces you have don't add up. And then you have to hypothesize biology. And then maybe the biology you hypothesize is actually something that there's data on. In that T cell example, I didn't originally have 3 states of resting, activated, and post-activated. I had said, okay, we know we need resting T cells, we know we need activated T cells. The dynamics didn't add up. I couldn't get the proliferation and the apoptosis to balance to give me the dynamics I needed. And so I said, I need a post-activated population. And knowing enough immunology, I know that population exists. For simplicity, I tried not to go there first. Later on, I said, okay, I have to, because I can't capture the behavior without it. And sometimes that's a case where you are pointed to a biology that does exist. Sometimes you'll be pointed to biology that you have no idea if it exists. You can say, I don't know how this is adding up. It could be there is a feedback from here to here. I don't know how that feedback works. And maybe that leads you to experiments to say, is there a feedback? How does this happen? And so it is very much an iterative process.
Thank you very much for your answer. Thank you very much.
So we need people like you. Working with us.
Yeah.
This is the reason
Yeah,
why
I
we
agree.
are collaborating with Seirin-san.
Yeah,
Thank you.
thank you. Thank you. Then we are going to the another general participants questions. So Tomoko Maeda Chubachi, if it is okay, you just turn on your microphone and you can directly give your questions to speaker.
Hi, can you hear me okay?
Yeah.
Okay, so thank you so much for a very comprehensive presentation. So my question is, with this advancement of this quantitative mathematical model and also the AI model, you can get a lot of data points from the phase 1 study and phase 2 study. as well as natural progression model as well. And with all data combined, don't you see that the future clinical trial, you don't need that, the placebo arm? Or how do you see that kind of future?
Yeah, there is a lot of work being done here to see if you can do virtual placebo arms. You have to have validated them very well for anybody to believe them, of course. You could use them internally to give you some ideas, but if you actually wanted to eliminate a placebo arm, you would need to have a very well-validated model. Right now, where you see more cases where that is used to, build the data beyond a limited data set, or to help design the trial relative to placebo, etc. But there are definitely cases where you can replace the placebo arm. And as you get more data, as you can develop better disease progression models, and that could be by AI, that can certainly add to that. There's whole companies working on this. There's a company called Unlearn AI, which is very much in the neurological disease space for these disease progression models, etc. Often they're helping with powering the study too. How much will AI specifically be able to build? I think we have to be a little careful in our expectations because you can have all of this data, but if you have to identify the right pieces of data, because you can have thousands of genes and thousands of expression and whatever, right? But then every patient looks different. So if you're talking about a placebo arm, what are the key aspects you're trying to capture? What is the common, a lot of that may turn out to be relevant. A lot of it may be very irrelevant. The other place I see this kind of rich biomarker clinical data from individual patients being very valuable is in rare diseases, where you cannot do large studies.
Right.
Every patient is almost its own study. And there, I think, having a systems model of the disease, which is tuned to the individual patients that you encounter so that you can explore the impact of a therapy, is perhaps a very valuable place. And you do see publications now more and more on, and especially because you have more and more treatments for rare diseases popping into the space of interest because of gene therapies and RNA therapies, etc. So I think that's another place where AI and digital twins and individualized biomarkers will be very helpful, is with rare disease.
Thank you very much.
So I would like to invite Professor Kawamoto for the question comment, please.
Okay. Thank you very much for your talk. It was very comprehensive and very much informative. And it was good for me to see that your QSP model can predict immune reaction. It's a very much complex system, but still you can predict what happens. So then my question is, so whether this QSP model can be used for our cell therapy. Yeah, you showed some of CAR T-cell therapy case, but in our case, we are using the cells that were generated from ES or iPS cells, pluripotent stem cells. Now we can compare these regenerated cells with original or peripheral T cells. We have such data, but so we have to determine how many cells to be given to the patient. And so far we are just referring to autologous T cell case, maybe 10 to 8 A's for per person or something. But if we can determine those of cells by this QSP modeling system, then it would be very much useful for us. But the point is our cells are a bit different from peripheral T cells. So then my question, do we have to start with animal model like mouse or primate to measure PK or PD, or can we just immediately use QSP modeling system? That is my question.
So if you were to come to me with that question right now, I worry about too much with the mouse and the cyno. Those are not great models for cell therapy, especially when you're talking about trying to translate. Is it better to use that with stem cell derived, or is autologous in human more informative? So one thing one could, for example, do is with these models of autologous therapy in humans, whether it's CAR T, which was the Curac paper, or TCR transgenic T, which is the Jocelyn paper. If you can look, and the Jocelyn paper very much looked at what should be the number of cells and the composition of cells, etc. But there were very specific parameters for those patients and for those cells, proliferation rates, things like that. If you could have hypotheses based on in vitro data or in vivo data, if it's better, of these cells proliferate more, these cells proliferate less, these cells differentiate more, these cells differentiate less. If you had that kind of quantitative data, one could start to say, okay, this was this system, but what if I dial the behaviors of the cell to look more like an iPSC, less like an autologous cell therapy? Then you could start to explore, oh, well, you know what? I could get away with fewer cells. Oh, well, I need a lot more cells. Whatever that finding is. But that would be how I would think about it. Especially, it could be different phenotypes. Your iPSCs may be far more stem cell-like. Your autologous may be far more differentiated. That one of those papers, the Jocelyn paper, looked heavily at which of the phenotypes, and the more stem cell-like T cells led to a better persistence of the cellular therapy. So.
Okay, thank you.
Okay, so since the time is limited, so let me, allow me to prioritize questions from advisors. So Dr. Kawai, please.
Yes. Saroja, thank you so much. Your excellent presentation. So I'm interested in how QSP and real-world data can be integrated for the drug development and/or in the post-approval space, because I think the real data is one big source for the drug development. So could you share your thoughts on the future direction for combining QSP and real-world data?
Yeah, I think that's really very interesting because so often, we've used data from clinical trials and there is this whole untapped for QSP body of if we can go into the medical records, get really, hey, there's a lot of data we don't have from the clinical trials. Okay, what if you followed this patient longer? What if you had this patient on a different co-medication? What if this patient, if we had that information, I think it could inform a lot more of our model development and our robustness of testing. That said, I'm going to actually suggest CJ is probably the better person to answer that question because whereas I have all these ideas and I got to work in the clinical space, I sat more and I tend to find myself more in the late research, preclinical, translational, early clinical, and occasionally in the later clinical. Whereas the CJ's QSP group at Pfizer did much more in the solid clinical, late clinical, and probably has thought a lot more about integrating those data. I have theoretical ideas. They've probably done it.
Okay. Thank you so much, suggestion. Thank you.
Okay, thank you very much. So it's time almost limited. So we are going to the final question to Professor Kabashima. Professor Kabashima.
Hello.
Okay.
Yes, thank you, Saroja, for wonderful and useful, informative lectures. I enjoyed a lot, but from my side, I actually have no more additional questions. Thank you very much.
But you're working in, you said skin immunology. So that's like after we worked in asthma, obviously there's a lot of relationship to atopic dermatitis. There's a lot of work in psoriatic arthritis, and there's just a lot of interesting work in that area. So hopefully it comes your way.
Yeah, thank you.
Thank you. Okay, so for the general participants, we have some questions. So we after this seminar, we just try to reply for your questions using via some emails. So okay, thank you for your participant. So Miyoshi-san, just
Okay, so then I'd like to close this session. Thank you, Dr. Ramanujan, your great talk, wonderful presentation. And also I'd like to thank the advisors and all the attendees for your very active Q&A session. It's really, really a wonderful time for us to learn and discuss the modeling. Thank you very much.
Thank you.
okay.
Arigato gozaimasu. And I really appreciate the invitation and the opportunity.
Okay, thank you. Okay, so thank you for every advisor, every participant and speakers and every steps. So we have another kickoff seminar in December. Please don't miss it. And thank you very much and have a good day. Good night.